Hi everyone, welcome to the only lecture that is in, I guess what we could call Unit 5. This is the only lecture that comes after Lecture Exam 4 and will be new material on the final. The final exam does cover everything from the entire semester, but this is the only new material that has not been covered on a previous exam. But more important than that, this is the last lecture of the semester. After this lecture, there is nothing else other than the final exam. So one last time, let's start with our not really attendance questions. First, what is a ganglion? Next, what is a nerve? And third, are you ready to not have any more of these questions? So go ahead and try to answer those on your own. And let's see what the answers are. So first, what is a ganglion? Remember, a ganglion is a cluster of neuronal cell bodies in the peripheral nervous system. There's something similar in the central nervous system called a nucleus, but a nucleus and a ganglion both are just clusters of neuron cell bodies. And in a ganglion, those cell bodies just happen to be in the peripheral nervous system. Next, what is a nerve? Similar to a ganglion, except instead of cell bodies, now we're looking at axons. A nerve is a collection or bundle of axons in the peripheral nervous system. In the central nervous system, we had some similar things called tracts, but a nerve is in the peripheral nervous system. And the third kind of goes without saying, are you ready to not have any more of these questions? I'm sure all of you answer that with an outstanding yes. However, if you have me for Bio 139, be ready because I do the exact same format for that class. So without wasting any more time, let's go ahead and dive in and get this one lecture out of the way. First, we're going to cover a little bit of info from chapter 13. And really, we're only covering one topic from that chapter, and this is the only topic from that chapter that will be on the final. And that's the structure of a nerve, which, as we just saw, is a collection of axons in the peripheral nervous system. And that's what we see here. So we can see here's the spinal cord. We don't see any vertebrae, but there would be vertebrae surrounding this. And here we can see the dorsal root ganglion, the ventral root, and here is a spinal nerve. So here is a nerve, and we're just going to look real quickly at the structure of this nerve and learn some terminology. And this terminology should sound very familiar. It's actually going to have the same basic layout as a muscle. Now, even though it's really common to kind of interchange the word nerve and neuron, remember they're not the same thing. A neuron is a single cell. A nerve is a collection of cell parts. So think back to when we were learning about muscles and we had fascicles and epimysium and perimysium and endomysium. Well, we're going to see the exact same thing here with this nerve, except instead of mysium, we're going to substitute the word neurium because we're talking about neurons. So here is a nerve, and we've cut it open so that we can see inside of it. Now, the whole nerve is surrounded by a connective tissue wrapping called the epineurium. So all this yellow that we see right here is the epineurium. And inside of that nerve, surrounded by that epineurium, we see bundles. One of them is pulled forward here, and this is called a fascicle. Each of these bundles is called a fascicle, exactly like we saw inside of a muscle cell. So these are fascicles. And then inside of these fascicles and between the fascicles, we see blood vessels. Now, each of these fascicles also has a connective tissue wrapping called a perineurium. So each fascicle is surrounded by the perineurium. All of the fascicles together are surrounded by the epineurium. Perineurium around the fascicles, epineurium around the entire nerve. But if we look at these fascicles, they've got smaller units inside of them still. And that's what we have pulled forward. Now these are the axons. So these are individual cell components. The axons, some of them are myelinated, some of them are unmyelinated. But regardless if they are myelinated or unmyelinated, they are going to have the axon that is surrounded by the cell membrane called the axolemma. We learned that back when we were studying neurons. The axolemma. The axolemma may or may not have a myelin sheath. And the myelin sheath, if it's there, will be surrounded by something called the endoneurium, which is another connective tissue sheath. Now the unmyelinated, here we see that it does not have an endoneurium. They could have an endoneurium, but they don't necessarily. The myelinated axons will have an endoneurium. So let's look at this myelinated axon and work our way up now until we get to the full nerve. So we have the axon, which has its own membrane called the axolemma. The axolemma is surrounded by the myelin sheath, which is surrounded by the endoneurium. Now we have a collection of those surrounded by the perineurium. Now we have a fascicle. We take several of those fascicles together, surround them by the epineurium, and now we have a nerve. Very similar structure to what we saw in the muscle. Okay, so that's all that we're looking at in chapter 13. Now we're going to jump ahead to chapter 16, and again, we're only going to look at a very, very small portion of this chapter. In chapter 16, we're going to look at sensory neurons. We're going to be looking at some sense organs. We're not going to be looking at the special senses like eyes and hearing from the ears or sight from the eyes, anything like that. We are just looking at basic senses. The sensory receptors come in several types. But first I want to talk about something that you actually did in lab this past week, or I guess recently, not this past week necessarily, but when you were doing the sensory lab. One of the tests that you did in the sensory lab was the 2-point discrimination test. And sometimes that can be a little tricky as far as what is it that you're actually testing for. So I want to kind of show you here with these images exactly what's happening in that test. Remember in the 2-point discrimination test, you took 2 pins or 2 needles or 2 of whatever, and you touched somewhere on your arm or your hand or your finger. And the first time you touched, you felt 2 points, and then you moved those a little closer and you touched, and you moved them a little closer and you touched, until eventually you could only feel 1 point, even though there were 2 of them touching. So why could you sometimes feel 2 and sometimes feel 1, even if there were 2 points being used? What it's actually testing for is to see how wide of an area is one neuron detecting. So here, this is like something in your shoulder blade, somewhere where you do not have really, really keen touch sense. So we can see one neuron, and then the sensory area of that neuron is spread over a really large area. And even though we are touching this with 2 points, we're stimulating the same neuron. So on the back of your shoulder, you could have 2 points pretty far apart, but it's only going to feel like 1 point. To demonstrate this, if you have someone that can do this for you, pull your shirt up so that your back of your shoulder is exposed, and have them take 2 pencils and tap like we see right here. And chances are you're only going to feel 1 point because that one neuron covers a really wide area. But in certain parts of your body where you need to have, kind of some discrimination, like we see here on the fingers, well, each neuron covers a very small area. So we could take those same 2 points and touch, and here we can see this one on the left is stimulating this neuron, this one on the right is stimulating this neuron, so we're going to feel 2 points. If we move them closer together, and this neuron pokes here, and let's say, this pin or whatever we're using pokes here, and this pin, let's say it pokes right here. They're really close together, but they're still going to be stimulating 2 neurons. So that's why in areas like your fingertips, we have a lot better sensation, so where we can discriminate really, really small things because we use our hands, we use our fingers for just that purpose. Different parts of the body, the neurons are going to have a much larger or much smaller area of sensation, depending on exactly how much discrimination we need for that area. So let's look at different types of sensory receptors. And we can classify sensory receptors a few different ways. One way that we can classify them is by what type of stimulus do they detect. And we can see a list here. We have things called mechanoreceptors. Mechanoreceptors detect things like pressure. And they do that because of something like physical deformation of the neuron itself. We talked about this a little bit back when we were doing different types of channels, like voltage-gated channels. We had mechanically gated channels. So mechanoreceptors respond to pressure of some sort. Next, thermoreceptors. Thermoreceptors respond to temperature, either hot or cold. Next, photoreceptors respond to light. This is what we have in the retina, the back part of our eye. Next, we have chemoreceptors. Chemoreceptors respond to various types of chemicals, and there's different types of chemoreceptors, and they're found throughout our body. But for example, both our sense of smell and our sense of taste are chemoreceptors. When we smell something, what we're actually doing is bringing air in from the environment, and there are chemicals in that air that will attach to and stimulate nerve endings deep inside our nose. Our taste buds are receptive to certain chemicals in whatever we put in our mouth. But we also have chemoreceptors throughout our body that detect things like pH or carbon dioxide levels, things like that. So different chemoreceptors will detect different types of chemicals. Next, nociceptors. Nociceptors are kind of sometimes just called pain receptors. Nociceptors detect things like damage or injury to tissues. When we damage a tissue, it releases certain chemicals, and those chemicals then stimulate nociceptors, which our brain interprets as, there's some sort of damage, let's make it feel like it hurts, so that we can draw attention to that area. Well, we could also classify the receptors by the location of the stimulus itself. For example, exteroceptors. Exteroceptors detect stimulus that has arisen from outside the body. So when you walk outside and it's cold, or when you look at a light and the bright light hits your eye, or when you breathe in and something smells like flowers. Well, all of those stimuli arose outside of your body. So those are all examples of exteroceptors. Interoceptors respond to stimuli that arose from inside your body. For example, if you have a stomach ache, or if you are trying to pop your knuckles and you pull it just a little bit too far. Things like that. So something that arose from inside your body. And the third type is kind of going to bridge the two. Proprioceptors. Proprioceptors are really detecting the position or movement of your body. This is how you know what position your body is in. So let's say you go into a room and there's a power failure and there's no windows in that room, so the room is completely pitch black. Well, even though you can't see yourself, you still know what position your body is in. You know where your arms are and if they're straight or bent. You know where your legs are and if they're straight or bent, because of these proprioceptors. And proprioceptors tend to be found in places like around your muscles, around your joints, things like that, because of how we're stimulating these proprioceptors. Our brain interprets that as, okay, my arm's in this position, my legs are in this position. That's proprioception. But when we're talking general senses, so general senses are not our special senses. Our special senses, like I said, that's sight and hearing and taste and smell, that kind of thing. So when we're talking general senses, let's look at the different types of sensory receptors. And we can classify them in two different ways mainly, and that's unencapsulated versus encapsulated. These three on top are unencapsulated endings. These 6 down at the bottom are encapsulated endings. The difference is the unencapsulated endings. The endings themselves do not have any sort of wrapping around them, whereas the encapsulated endings, the sensory part of the neuron, has some sort of either connective tissue or glial cell surrounding the receptive part. So first, let's look at the 3 unencapsulated nerve endings. First, we have free nerve endings. Free nerve endings are just completely exposed. We tend to find them in epithelial tissue, in connective tissue, but they could be anywhere in the body. And we can see the nerve endings just kind of reach out in between cells throughout the tissue. They detect things like heat, like cold, like pain. That's free nerve endings. The next one is a tactile disc or a Merkel disc. We talked about this back in skin. We find these right where the stratum basale of the epidermis meets the dermis. So the tactile disc, remember, it is not itself a neuron. It conveys information to the neuron. But what do we detect? Well, we detect things like very, very light touch. It doesn't take a lot to stimulate these guys. And the third type of unencapsulated nerve ending is a hair receptor. So again, we're looking at the skin. We can see the neuron comes up, and then the sensory part of this neuron surrounds this hair follicle. And when something moves this hair, it's going to slightly move this hair follicle. And that's why even if you don't touch your skin itself, like the surface of your skin, if there's, say, some dead skin or a bug or something that only brushes against your hair, or if you look down at your arm and you just brush the hair on your arm without touching your skin surface, you can still feel it. Well, hair itself does not have any neurons, nerves at all in it. So it's not sensory. What you're actually doing is stimulating these hair receptors by moving the follicle a little bit. Now, these final 6 are encapsulated endings. So the endings, the receptive endings of these neurons are wrapped in connective tissue or have associated glial cells with them. First, we see a tactile corpuscle. We find these in the dermal papillae. We find them in areas that are really, really sensitive, like the lips, the genitals, the nipples, the tips of the fingers. They detect very, very light touch, very, very light pressure. These were also what we called Meissner's corpuscles back when we were studying the skin. Here we have an end bulb. End bulbs are practically the same thing as these tactile corpuscles. End bulbs, though, we find these in mucous membranes. Next, we have bulbous corpuscles. Bulbous corpuscles are a little bit deeper. We find them in the dermis, we find them in subcutaneous tissue, but especially we find them in joint capsules. And here we have a sensation of firmer pressure that goes on for a long time. So it's pressure, but not just quick pressure. It's pressure that is sustained. So it's long touch. It also detects joint movement. So the movement of a joint changes the amount of pressure on this corpuscle. Next, we have lamellar corpuscles. We saw these in the skin also. We called these Pacinian corpuscles. So lamellar or Pacinian corpuscles we find in the dermis, like we saw in the skin, but we also find them in joint capsules. We find them in genitals. We find them in the breasts. And they are responsive to deep pressure. So not light touch. This is a deep pressure. But also they're responsive to stretch. They're responsive to vibration. Also, when you tickle someone, these are what are being stimulated. Next, muscle spindles. Sometimes these are called spindle fibers, but muscle spindles surround skeletal muscles. They tend to be at the areas close to a tendon, and they're responsive to stretch of that muscle. And the way they do that is we can see here they are kind of wrapped around the muscle cells. And when the muscle cell is stretched, the muscle cell is very thin. But when the muscle is unstretched, the muscle cell is a little wider. So they're detecting how wide or skinny a muscle cell is. And then based on that, they could tell how stretched the muscle cell is. This prevents you from overstretching your muscle, and it conveys information, again, about the position of your body. And the last one we see is a tendon organ. Sometimes these are called Golgi tendon organs. And we find these wrapped around and across tendons, very similar to muscle spindles, but these are on the tendons themselves. and they detect the tension on that tendon. And based on how much tension there is, it tells how stretched that tendon is. And just like the muscle spindles, it conveys information about the position of your body. So now we're going to spend the rest of this lecture talking about the autonomic nervous system. Up to this point, most of what we've been talking about are either the central nervous system itself, or we've been talking about the motor division of the nervous system. This lecture, we've been talking about the peripheral nervous system as far as sensation, and now we're going to talk about the autonomic nervous system. Unlike when we were talking about skeletal muscles, when we're talking about the autonomic nervous system, this is all going to be involuntary responses. This is the stuff that keeps you alive without you having to think about it. And we briefly mentioned this at the start of our nervous tissue chapter. But this table shows us the difference in the somatic versus the autonomic nervous system. But real quick, let's do a brief overview of ganglion and nerve, just to make sure everyone is still remembering exactly what these terms mean. When we're talking the autonomic nervous system, we're going to be looking at different nerves, where they are in the body, and what they are releasing as far as neurotransmitters. There's going to be one neuron called the preganglionic neuron. Its cell body, or soma, is somewhere in the central nervous system. And then it's going to have axons wrapped as a nerve extending to the peripheral nervous system, where there's going to be a cluster of cell bodies. I've got one cell body drawn here, but remember, this is a ganglion, so there would be a cluster of cell bodies. So this ganglion, somewhere in the peripheral nervous system. And then the axons from those neurons as a nerve are going to travel through the peripheral nervous system to an organ. This nerve is called postganglionic. So we have preganglionic nerve and neurons, postganglionic nerve and neurons. The soma of the preganglionic will be found in the central nervous system. The soma of the postganglionic are in the ganglion itself, somewhere in the peripheral nervous system. And this is going to be the layout for the autonomic nervous system. So here let's look at some specifics. When we've been talking about nerves and what their effectors are up to this point, we've been talking about the somatic nervous system, things that control skeletal muscles. So here we have one single myelinated neuron that goes from the central nervous system all the way out to the skeletal muscle, the effector organ. And this one single neuron or nerve with neurons inside of it releases acetylcholine onto that muscle. But that's different when we look at the autonomic nervous system, and that's what we see down here. In the sympathetic nervous system, we're going to have a neuron that goes to a ganglion, and then another neuron that goes from the ganglion to the organ. Same thing down here in the parasympathetic nervous system. A neuron that goes to a ganglion, and then a neuron that goes from the ganglion to the organ. The organs in this case are going to be organs that we do not consciously think about moving. Here it shows us the heart. It could be any number of organs. So let's look at a couple of differences in the sympathetic nervous system versus the parasympathetic nervous system. First, we're going to look at the function of each. When we're talking about the sympathetic nervous system, the SNS, we said back when we were talking about nervous tissue, we said that the SNS is responsible for our fight or flight response. Our fight or flight response. What do we do when we are under some sort of stress? This does things like increases our heart rate, increases our breathing rate, increases our blood pressure. When we're talking about the parasympathetic nervous system, the PNS, the parasympathetic nervous system we can sum up as rest and digest, or relax and reproduce. So rest and digest, this is the part of our nervous system that is in control when we are really mellow, when we're just sitting on the couch. This does things like slows down our breathing, slows down our heart rate, drops our blood pressure, moves food through our digestive tract. So just to kind of show you how one of these things is always in control and the other is kind of turned off, here's an example. Let's say you're sitting at home watching TV, laid back, eating some popcorn. Well, during that time, your parasympathetic nervous system is turned on and your sympathetic nervous system is turned off. We're in rest and digest mode. You're breathing slow, you're digesting that popcorn, you're super relaxed. But then all of a sudden, someone kicks your front door in and starts yelling at you and points a gun at you. Well, you're probably not so relaxed anymore. Your parasympathetic nervous system turns off and your sympathetic nervous system turns on. Your sympathetic nervous system, your fight or flight response, now you're going to either be ready to fight for your life or run for your life. You're breathing fast, your muscles are tense, your heart rate goes up, your blood pressure spikes. This is your SNS taking over. Hours later, assuming everything turns out all right, you're going to start to calm back down. Your blood pressure is going to come down, your heart rate's going to come down, your breathing rate's going to come back down as your sympathetic nervous system starts to turn off and your parasympathetic nervous system starts to take over again. So one of these is always on and one is always off. And depending on the situation, that tells you which one is on or off. Now that was a pretty dramatic example. They're not always going to be that dramatic. There are situations that aren't super stressful, but that will still kick in your sympathetic nervous system. For example, when you exercise, your sympathetic takes over. If you drink a lot of caffeine, your sympathetic will kind of turn up a little bit. So that's the difference in functions of the sympathetic versus the parasympathetic. But let's look at the anatomical layout. And what I mean is, where is the pre-ganglionic neuron? And where is the postganglionic neuron? And where is the ganglion itself? And where do these nerves leave the central nervous system? So first, let's look here at this middle picture. This is the sympathetic nervous system layout. Well, in the sympathetic nervous system, the ganglion is going to be right next to the spinal cord. It's right next to the spinal cord. So The preganglionic neuron is going to be really short. It's going to have a cell body somewhere in the spine, and that neuron, well, the whole nerve really, is going to go from the spine to right next to the spine, where we find this ganglion. So the preganglionic neuron is very short. But now, once we get to the ganglion, we have this next cell body. The nerve going from the ganglion to the organ, so the postganglionic fiber, is going to be really long. It has to go right next to the spinal cord all the way to the organ that it innervates. So what does that mean? The preganglionic sympathetic neuron is very short. The ganglion is right next to the spinal cord. The postganglionic neuron is very long. Preganglionic neuron, very short. Ganglion, right next to the spinal cord. Postganglionic, very long. If we look down here at the parasympathetic nervous system, the preganglionic neuron has a cell body somewhere in the central nervous system. The ganglion is on the surface of the organ that we're innervating. So the preganglionic neuron is very long. Then we get to the ganglion. We have a cell body there that just has to go from that ganglion on the organ to the organ itself. So the postganglionic neuron is very short. Parasympathetic nervous system, very long preganglionic neuron. Ganglion is on the surface of the organ that it is innervating. Very short postganglionic neuron. Now, what is released from these neurons? When we looked at the somatic nervous system, it always released acetylcholine, ACH. When we look at let's do parasympathetic nervous system. When we look at the parasympathetic nervous system, rest and digest, the preganglionic neuron releases acetylcholine onto the postganglionic neuron. The postganglionic neuron releases acetylcholine onto the organ, which then has those effects that we were just talking about. It slows down your heart rate. It helps move food through your digestive system. It slows down your breathing rate. All of the stuff that we think of when we're relaxed is because the postganglionic neuron is releasing acetylcholine onto our organs. Now we go to the sympathetic nervous system. In the sympathetic nervous system, the preganglionic neuron releases acetylcholine onto the postganglionic neuron. So everything so far has all been acetylcholine. Now we're about to change that. The postganglionic neuron releases something called norepinephrine. Norepinephrine. It releases the norepinephrine onto the organ that it's innervating. And the norepinephrine has the effects that we think of when we're talking fight or flight. It speeds up your heart rate. It makes your heart beat harder. It causes your blood vessels to constrict. It causes you to breathe faster. So everything in the somatic is acetylcholine. Everything now when we're talking about the autonomic nervous system, everything in the parasympathetic, the preganglionic, and postganglionic is also acetylcholine. When we talk sympathetic nervous system, the preganglionic is acetylcholine, the postganglionic is norepinephrine. So let's think about one difference with the sympathetic nervous system. There is also something called epinephrine, and epinephrine kind of continues your fight or flight response. When we looked at this, when the norepinephrine is released onto the surface of your heart, it has a very quick effect. But that effect does not last for very long. When we think about when we're really scared, we have that immediate burst of energy. Let's think you walk home and your roommate jumps out from behind the door and scares you. Well, you get that very instantaneous, you lock up and you're ready to fight someone like we see right here. And that's really quick. That's the norepinephrine. But then for several minutes after that, you've got that kind of nervous, tensed up feeling. That's not the norepinephrine at that point. That's epinephrine. So let's see how epinephrine works. When we're looking at the sympathetic nervous system, which is what we see over here in green, we have the neurons exiting the spine and going to a ganglion and then going to the organ. But there's one difference. Some of those neurons, instead of going to a ganglion, they go to an adrenal gland. And the adrenal gland is in place of that ganglion. Let's go back to our picture here. In one part of the sympathetic nervous system, we have a neuron that comes out and there is no ganglion. Here, where this ganglion is, we have an adrenal gland. And the preganglionic neuron releases acetylcholine onto the adrenal gland. The adrenal gland is part of your endocrine system and your sympathetic nervous system. So what happens when acetylcholine is released onto your adrenal gland? It starts to release something called epinephrine and norepinephrine into your bloodstream. About 80% of that is epinephrine, about 20% is norepinephrine. Collectively, they have another name that you've probably heard of. They are called adrenaline. Adrenaline is released from the adrenal gland. This then goes into your bloodstream. So now we just got that real quick, instantaneous response from the norepinephrine from the postganglionic neurons. But that sustained feeling of nervousness comes from the adrenaline, the epi and norepi, that's circulating in your blood. And that keeps that sensation going for quite a while. So, SNS, fight or flight response. We've just talked about the anatomical layout. In the case of the postganglionic neuron, it releases norepinephrine, NE. In the case of the adrenal gland, the adrenal releases epi and norepi, collectively called adrenaline, into your bloodstream that has the sustained response. Here's just to kind of demonstrate what I was talking about. Here's the spinal cord. Here we do see a vertebrae. But right next to the spinal cord, there's these things called the sympathetic chain ganglia. Here's one. Here's one. Here's one. These are those ganglia right next to the spinal cord that I was talking about. But it's not the entire spinal cord. When we look at this, the sympathetic nervous system comes from this green area right here. The sympathetic nervous system comes from your thoracic vertebrae and the upper part of your lumbar vertebrae. It comes from T1 to L2. T1 to L2. These guys right here. When we talk about your parasympathetic nervous system, the rest and digest, that they all release acetylcholine. They also don't come from your entire spinal cord. Some of them come from your brainstem, up here where we see the purple. Some of them come from your sacrum, down here where we see the yellow. And remember, the parasympathetic nervous system has long pre-ganglionic, so it's going from the brainstem all the way to the organ, or from the sacrum all the way to the organ. The post-ganglionic is very short because it only has to go from the ganglion on the organ itself to the organ. And here's just kind of a summation of sympathetic versus parasympathetic. Where does it leave the central nervous system? Where are the ganglia? How long are the preganglionic? How long are the postganglionic? What's the effect of those? And here is a more detailed, I do not expect you to know these. This is just to kind of give you an idea of what does the sympathetic versus what does the parasympathetic do at different parts of the body. All right, so that is it for this lecture. That is it for this unit because there's only one lecture in this unit. Remember, that doesn't mean that only this lecture is on the final. No, the final covers everything from the entire semester. This is just the only new stuff that will be on the final. So you have officially made it to the end of Bio 137. Now, just to give you guys my end of the year talk that I always give, it's easier when it's in person, but just because of it being online, I still want to give the end of the year talk. And that is going to be that I am now officially done as far as teaching the class to you. But it is up to you when you want me to stop being your teacher. I will be your teacher as long as you want me to be. If you never want to hear from me again, that is entirely up to you, and you don't have to after this grades post. But if you are ever in Bio 139 with me or anyone else, when you move into your programs, when you move into your career, if you ever have a question, please don't hesitate to ask. I'm as willing to respond to texts or emails 5 years, 10 years from now as I am all this semester. I don't ever want to lose touch with you if you want to stay in touch with me. I love hearing from old students. I'm to this day hearing from students, even from my very first semester, telling me awesome things that happened to them when they were with a patient. Please don't break HIPAA guidelines. Don't tell me anything important about a patient's identity. But if you want to tell me something that happened in the operating room, man, I would love to hear that. If you have things that you think would make a good case study, I'm always looking for good real-world examples of case studies. But I am here to help you as long as you want me to be here to help you. I am so sorry that I don't get to meet each of you like I would in an in-person class. Now, the other thing that I want to tell you is most of you are going to go on to take Bio 139. I know this was a tough class, and I know you are ready to get it behind you. But please, please don't flush this information now that you're done with the class. Most of what we have learned is going to be foundational for Bio 139, especially the last part of this lecture. Almost every single day in Bio 139, you're going to talk about the SNS and the PNS, and it is not going to be reviewed. It is assumed that if you got through Bio 137, you completely understand the material in Bio 137, and they are not going to review it. Bio 139 is really just the second half of this textbook. It's not a deeper look at what we've already done. So for those of you that would like to get a head start in Bio 139, here are the things that you really, really want to make sure you understand. And I'll kind of go through in order of what you want to review. You want to understand what ions are. You want to understand pH and concentration gradients. You want to understand action potentials. You want to understand muscle contraction and things that influence muscle contraction. And you want to understand the autonomic nervous system. Those things are foundational for Bio 139. If you have questions over breaks, I'm always happy to help. If you have questions on weekends, I'm always happy to help. Just text me or email me, and I'll be there. All right? Don't turn off your brain just yet. You've got the final to go. Most students tend to do pretty well on my final. They've gotten this far. They've got the hard stuff behind them. Now it's just a matter of recall. Let me know if you have any questions. Take care, and please let me hear from you when you have those good stories about your success. Have a good afternoon.